Fantastic.
Yeah.
All right. Well, look, I think we might just get started. I think of the presenters, it's only Merrin that we don't have here.
Here, Merrin is here.
Merrin's here. Excellent. Okay. So, look, we'll just get going. In Darius's absence, very kindly, Hasib has agreed to chair the meeting, so I might just hand it over. Thanks, Hasib.
Thank you, Peter. Good evening to you all, and welcome to the 2025 AGM for the Australian Amyloidosis Network. To get things started, I'll do a welcome to country. So we acknowledge the traditional custodians of the country throughout Australia, wherever we're all meeting in the different corners of Australia, and pay our respects to elders past, present, and emerging.
Yes.
To begin proceedings, I would like to propose confirmation of the minutes of the AGM from 2024. Do we have anyone that's willing to second that? I see Pat's hand up. I think that's all we need. So we can confirm those minutes. Thank you, Pat. First item on the agenda is the chairman's report. So I'd like to welcome Peter to take over and present us with the 2025 chairman's report.
Thanks, Hasib. Let me just try and get my mouse working. All right, can I check? Can you see that screen? Thanks. Can I just, if anyone hasn't muted themselves, can you just mute yourself? There's just a little background coming through every now and again. So anyway.
Sorry, Peter, are you sharing the agenda?
No, I'm sharing my talk.
In that case, I cannot see that. We've still got, I think, the agenda, which I'm assuming might be shared.
Get in there.
Okay.
I've stopped that share now.
Thanks, Libby. I'll go again. Okay, you got that one?
Yep.
Yes.
All right. So, look, my summary of the last year's activity has been circulated already, but I'll just run through a few things for everyone. So just a little bit of history. The AAN sort of existed unofficially for a number of years, but we incorporated as a company in December 2021. So we've been around for almost 4 years now. We got our charitable status the following year. And here you can see the current board members. And I really want to pay tribute to and thank the board members of the AAN. As opposed to many boards, it is a very hands-on board. All the members end up doing a lot of work on behalf of the organisation, and we just couldn't do half the things we could without everyone contributing. So you can see there Kath Davies, who came on just before our AGM last year as our patient advocate, and Kath has been a real great boon to the board. Merrin, who brings her business skills and particularly her financial skills. We couldn't function without her. I certainly know nothing about finance, so it's been terrific to have Merrin on board. Peter Bridgman, who got particular governance expertise, and his legal background is really important. There's a lot of contract review that go on, a lot of information that has to be reviewed to make sure we're compliant with various bits of legislation. And the two other medical members of the board joining me, Hasib Siddiqui, who heads the Fiona Stanley Amyloidosis Service, and Fiona Kwok, who heads the Westmead Amyloidosis Service. I'd like to give a particular shout out to Dr. Olga Matorna, for the last couple of years has been acting as the alternate director for Simon Gibbs and has done a fantastic job. As you'll hear later tonight, made some particular contributions. This year, with Simon taking a permanent position with Pharma, he's had to step down as per the Constitution. And so because of that, Olga has stepped down as well, and we'll be looking to boost some of the skills and members on the board in the next year as well to replace those. We've also got Darius Korchik as our company secretary. As you mentioned, he's an apology for at the moment. And Libby, our executive assistant. I'd like to make particular mention of Libby. I mean, Libby works well beyond her paid hours. She's completely dedicated to this organisation, does so much work for the board and for the other committees, shuffles around between different people, sorting things out, trying to solve problems. So thanks very much for all your work in the last year, Libby. We just could not function without you. All right. The AAN has got a lot of committees. Sometimes I think we've got more committees than members, but that's not true. But we've got a lot of things we want to get done, and we need people to do that. So you can see under the board, we've got listed here the Governance Committee, Partnerships and Philanthropy, Finance, Nominations, Advocacy and Research, the Cardiology Committee, Media Communications, Membership Committee, and not a formal committee at the moment, but the working group that organised the workshops. And they're with a special place, I guess, because the unique nature of that committee is the Patients and Family Committee led by Gary and Maureen, with direct input into the board. And I'll mention briefly in a minute, but our newest employee, our telephone support manager, Jess Schwindiger, who's joined us as well. So all of these committees have done a lot of work. I'll take you through some of what they've been have done in the last year. And my apologies in advance. I can't mention everything that everyone's done. So I've missed out. If I miss out something that's important, I apologise for that. But there really has been an enormous amount of work. I think we have been busier this year than ever before. So I'll start with Patients and Families Committee briefly, chaired by Gary and Maureen as interim chairs. They've been interim for quite some time, but they continue to chair the committee, leading a number of activities, and I'm only going to mention a few. The first thing that happened in the last year is this project of John Main's developing an amyloid patient information sheet, which is available on the website for patients or families, their family members to download. A tool meant to improve communication between patients and their healthcare practitioners, assist them if they have to seek urgent medical care somewhere else, that they can have all their history, their doctors, their medications, for example, available. So hopefully that'll be a useful tool, and we'll be looking to get some feedback on how it's used in the next year. Kath has introduced a couple with Kath, which is a virtual support group where Kath runs a session, as it says there, looking at the ups and downs, the ins and outs, and all things amyloidosis. And that's been well attended, and we've had a lot of good feedback about this. So thanks very much, Kath, for running that. It's not always easy to get all these things organised, but that's been a great addition to the support the AAN can provide. This year, under the guidance of Susan Booth, we joined the International Amyloid Survey run by the Amyloidosis Alliance, which is a major worldwide survey looking at the needs of patients with amyloidosis. And we got very close to our required 200 patients and families to complete that survey. That's being analysed, and hopefully before too long we'll be able to see the data from our Australian contingent. And that'll be very informative to help try and plan what we need to do over the coming years. So hopefully next year we'll be able to share some of that with you. Kath also attended the Amyloidosis Alliance. You might have heard us chatting about that earlier on. And we'll be getting some feedback from Kath at the board level and no doubt in the various committees as well. I think it's very important for us to interact with the other amyloidosis groups around the world. We do that scientifically through a number of the medical meetings, but this is an important way to interact with other patient groups as well. I mentioned the appointment of Jess Schwendinger. Jess resides in Perth and was appointed only a month or so ago as our telephone support manager to design and then implement a telephone support service for the AAN. And this is going to be a very important front-facing part of the AAN, a way to provide psycho-emotional support, some health navigation, basic information, helping people get into the materials they need and providing patients with the right questions to ask. So Jess is going to make a first report to the board next week, in fact, so we're going to very much look forward to the development and rollout of this service. Support not going to end there, though. We need to build up our website support, something that's on the board's mind, but we haven't introduced yet, is to improve face-to-face support meeting, partnering with the various amyloidosis centres in the States. And this is going to be something important to progress in the next year. The membership committee is chaired by Angie Yong. The AAN is growing really at a surprising rate, I think. We now have over 400 members, more than 100 than last year. And there's 131 health professionals and 316 patients and families, and our 2 emeritus members. John Main produces a regular newsletter for the patients and families, and Angie one for the healthcare professionals. And I think actually this year's newsletters have been the best we've seen. They've really been very professionally produced and very informative. So I commend those 2 people for the work they put into those newsletters. That's important for our members. This year, of course, has been a little bit dominated by the workshops, which were held in 5 different cities: Perth, Adelaide, Melbourne, Sydney and Brisbane. So this is a major undertaking by the AAN, and this year's workshops were chaired, or the committee was chaired, by Fiona Kwok. Probably added a few years of life to her, or took a few years away, whichever way it goes, certainly a few grey hairs. But anyway, ended up being a terrific series of meetings. Our highlight, of course, was our international speakers, 2 in person and 1 online. Carol Whelan from the UK National Amyloidosis, one of the world's leading cardiologists in terms of amyloidosis. Stasis Kastridis, a haematologist from Greece. Again, for those who follow the medical literature, the first author often on many of the world-leading trials in amyloidosis. And from Japan, Professor Yoshi Sekijima, one of the most qualified doctors I've ever met. He's qualified in 3 different specialties. So that was terrific to see those meetings in 5 cities. More than 340 patient and family attendees. The reason I've got plus is we're not exactly sure how many attended through the webinars. 320 healthcare professional attendees, 11 pharma sponsors. So really an enormous amount of work, but a major contribution to the education of healthcare professionals in Australia in regard to amyloidosis. And of course, a great opportunity for patients and their families to learn more about their disease. We introduced another couple of educational initiatives this year, organised by Kath Davies and Olga Matorna. These are 3 seminars that were held virtually. The first one in April with Darren Lee, a nephrologist, and Mahima Kapoor, a neurologist. The next one at the end of July with Daniel Chen, a cardiologist, and Margaret Ryan, a clinical nurse practitioner. And then celebrating World Amyloidosis Day, another seminar featuring Pat Neely, Hasib Siddiqui, and Christina Ayres. And you can see the increasing representation there from the various amyloidosis centres around the country. And those seminars have been very well attended, particularly that last one on World Amyloidosis Day. I think we had 40 or 50 attendees, so that was terrific. So thanks for Kath and Olga for organising that. Also, towards the end of this last year, we had our first educational episode targeting GPs. Dr. Yap, one of our members in Sydney, who also has lived experience with amyloidosis, wrote an article for Australian Doc. Various AAN members have presented at haematology, cardiology and nephrology Australian scientific meetings, and we've run a couple of seminars run via the Limbic or United Clinical. The last one allowing the webinar to be made available to the AAN to put on our website, and also the last one making a donation to the AAN as well, which is fantastic. The Cardiology Committee, led by Lisa Thomas, has been busy as well. Last year, they published the consensus paper on cardiac amyloidosis management, and also published a snapshot of ATTR amyloidosis in Australia. They ran an educational event at the scientific meeting in Brisbane, and importantly, have managed to raise significant funds to enable the creation and maintenance of a transthyretin amyloidosis registry. And in fact, we just sent off the invoices today to get the funds to start that up. So that's been a long-term project, something we've wanted in our strategic plan. So that's going to be fantastic to actually get up and go. I was just having a little look at what our AAN members have been publishing in the last year. And in fact, there were a lot of publications. I've just pasted them all in. These are not all of them, and I apologise if your publication's not there. But gee, there is a lot of research activity going on in the area of amyloidosis by Australians. And this was terrific to see across the breadth of basic science, clinical, registry, position or guidance statements. Really, we're contributing a lot to research worldwide. The AAN Partnerships and Philanthropy Committee was a really important initiative. This committee was inaugurated by Dr. Simon Gibbs just at the beginning of this year, when Simon returned from overseas. Simon had to step down when he took his full-time pharma position, but has handed over to the current chair, Keith Moore, who is doing a cracking job at trying to continue the work of Simon. Simon, as you know, is a tornado of ideas and activity, and certainly we saw a marked increase in donations in the last year, over 100,000, which is just fantastic. We have got a lot that we need funding to support, and this committee is a key one. There was a lot of fundraising at the workshop. We had a successful end of financial year appeal. Keith recently ran a quiz night in South Australia that raised several $1,000s, and there was a rowing challenge where a group provided a quarter of their fundraising to the AAN, and that was just only held last week. Very successful. So we really appreciate that assistance, and we've got a Christmas appeal coming up. So this committee, if they reach out to you, respond enthusiastically. Their activity is going to be key to the financial sustainability of the AAN. The Media and Communications Committee, led by Pat Neely and Tavita, has also been extraordinarily busy. I think this is perhaps the busiest subcommittee on the AAN. The website has undergone a major reskin. It was done in time for the workshops, particularly complete overhaul of the medical information, which was fantastic. We get data on how many hits this website has, and it is a surprisingly large amount, both from within Australia and in fact internationally. So a lot of people are coming to this site for information. And these are not just people who just click on it and move on. These are people who are spending a significant amount of time on the website. There's regular updates on clinical trials. There's regular updates on events. And coming shortly is a revamp of the patient and family information section. So look out for that. So thanks very much to that committee. There's really a large amount of work that's done. And to our web designer, Don, who also puts an awful lot of work into trying to achieve what we want on the website. The Advocacy and Research Committee, there's only been one drug that's come out of the PBS this year. That's vutrisiran for hereditary ATTR amyloidosis with neuropathy. It's got a list price of $450,000 per year for this extraordinarily expensive drug, but approved by the government because of its effectiveness. And it's wonderful to have therapy available for this group of patients. BNP came on the MBS for once-a-year testing. That we had nothing to do with that, but the Cardiac Society put that. But we are working with them to enable funded availability to increase frequency of this testing, which is critical for monitoring cardiac amyloidosis. And is not available at the moment. So patients in the private sector have to pay for this, and that impedes good medical care. So we'll see how we go, but hopefully we might get somewhere with getting this testing funded. Courtesy of Joanne Reid, we also had our first dedicated meeting for laboratory scientists in concert with the Sydney workshop. And in fact, we've just a couple of days ago got funding support confirmed to hold a full day laboratory and clinical research event in Sydney, which will try and time around World Amyloidosis Day next year. So this is going to be great. It's been a key goal of the Advocacy and Research Committee to try and improve particularly the laboratory and translational research in amyloidosis in Australia. So thanks to Joanne, and also for Natasha, who's done Natasha Gorry, done a lot of the research, the work to try and support this research workshop next year. There's a lot of trials going on in amyloidosis. I've listed some in AL amyloidosis and TTR amyloidosis there, as well as some basic science research, which we'll hear from our guest speaker later in the meeting. I'll just finish to remind you of our strategic plan. We have got a vision which is working together. This is extremely important. The AAN is an unusual group in the health not-for-profit area. Most groups are either patients-based or physician-based sort of professional societies, but we had a vision many years ago to set it up as a group of people working together. And we believe that patients and healthcare professionals working together is the quickest way to try and improve outcomes for patients. And seeking a cure, because that's what we really want. We don't really want to just make people feel better. Improve quality of life, very important. But really we want to try and get rid of this terrible disease. And there's been enough improvement in the last 10 years to think that this may actually be possible. Our values of respect, compassion, integrity, innovation, and collaboration are very important. It's not always easy to collaborate together with many different people from different walks of life, but it's very important if we're really going to get somewhere. We've got our strategic pillars of improving patient outcomes, building knowledge, financial sustainability, and organisational excellence. We'll be revisiting the strategic plan later on next year, but it's the foundation of our company. That's all I've got. Thanks very much for the opportunity to update you. Thanks, Haseeb.
Thank you so much, Peter, for that fantastic report. And I think one thing I can say is, based on the yearly reports over the last couple of years, just to see how much just how much work is being done through the AAN year by year, and how much that activity is increasing is a fantastic testament to everyone who's putting the work in. So thank you for that report. I think we will have an opportunity for people to ask questions from the board of directors at the end. So I might leave questions for the end and move to accept the chair's report. Can I have someone seconding that? I've got Kath there who's got her hands up, so that motion can be passed, Libby. And next item on the agenda is the Treasurer's report. And it's my pleasure to welcome our Finance Director, Merrin Brookman, who has put in a lot of work through the Finance Committee to ensure that we run a tight ship. So I know you're struggling a little with some IT issues, Merrin, but we'll see how you go. I'll let you take sharing.
Thanks, Hasib. Can you hear me okay?
Yes.
Thank you. And Libby, can I just share my screen, please? Okay. Can you see that okay?
Just about, yes.
Yes.
Perfect. We've got it on.
Great, thank you. So hello everybody, I'm Merrin. I am a member of the board and I primarily look after the financials for the organisation. Sorry, please bear with me, I'm just having a couple of tech issues. Okay, so as outlined by Peter, we've had a really big year at the AAN with the workshops, with the establishment or the hiring of our telephone support manager and the imminent establishment of that facility. And really we've had a really big uptick in the generosity of our donors and our sponsors this year. So as you can see, we've had sponsorship funds received year on year has increased dramatically, as is always the case because we had our workshops this year. However, this was the year that we received the most in our sponsorship funds from pharma. So we've had a really fantastic year in that regard, 440% up year on year. Donations, the generous donations provided primarily from our patients and their carers, we've seen a 74% increase year on year from the donations. So that's really was driven by the establishment of the Partnership and Philanthropy Committee and the fabulous work that Simon and Keith and everyone on the committee has done. We're hoping to see that continue throughout this year. Our membership funds is increased by 50% also, which is wonderful to see, and it's really evident that we're growing our membership base. And our registration funds were increased 100% this year because we had our workshops. So at the end of 30 June 2025, which is the financial information that we're presenting in this meeting, we had $399,722 in our bank accounts. So that really sets us up to be able to really progress our projects and achieve our outcomes for primarily the patient support, telephone support program and the ATTR registry. The next slide is a little bit heavy on data, but I just thought it would be important to provide you with a full financial outlook. So you can see there, probably the main things to draw your attention to is our major expenses. So our media and communication, we invest heavily into our website and our communication with our members, which is fundamental to what we're trying to achieve. The other major expense comes from the workshops that were undertaken, of course, and our wages and salaries for Libby, the wonderful Libby, and now in this current financial year we've been joined by Jess. As I said, $400,000 in our bank account at year end. We had a small receivable, which was an amount owed to us from the organiser of our workshops. We have a small amount of office equipment. This year we did have to register for GST, so we have a small amount of GST outstanding and some payroll accounts. And our current earnings for the year was $80,000. So that's really all from me, but I'm very happy to take any questions later on during the question section.
That's fantastic, Maren. Thank you so much. Yeah, I think we might just leave the questions to the end. Just from my end specifically, thank you so much, Maren, for all your help in managing the finances. I am the treasurer, but I feel like you've made my job a hell of a lot easier and simpler. So thank you for that, and thanks for the finance report. So I will move to accept the finance report for 2025. And I have someone seconding that. Yes, Kathy, thank you so much. So we will pass that motion. And next item on the agenda, which I think was an important step for us, was the appointment of an auditor. So there is a resolution that has been proposed that Diana Taylor, CPA, be appointed as the Australian Amyloidosis Network's auditor for the next reporting period. Maren, did you have any comments about appointment of Diana as our auditor?
Only that we have used Diana now for the last 2 years, and she's been really fantastic to work with, and is actually specialises in the space of not-for-profits. So I would certainly recommend that we pass this motion.
Fantastic. And do we have anyone else seconding that? I can see Peter's hand up. So we will pass that resolution and appoint Diana as our auditor. Okay, and now I think next item on the agenda, or we have a few moments for any questions that anyone on the line might have to any of the directors on the board. So if you'd like to ask a question, maybe the best way to do it is to use the raise hand function, and I can call upon you to ask your questions. Keith.
G'day everyone. Everyone here who doesn't know me, I'm the Director of the Partnerships and Philanthropy Committee. I do have a question, just with the expenditure of the workshops, based on previous years, why this year we spent so much more on the workshops, and why so much more was spent in the eastern states compared to the rest of the states?
I'm probably best to answer that. Yeah, so we had more people in the eastern states. The eastern states also, so for instance, venue hire is usually the majority of the funds, and it's per seating and per space. And we did actually try and keep costs down, but some of the eastern states in particular are very high cost. And I would say that we actually haven't spent more in comparison to the last workshop 2 years ago.
And if I can just make a comment on that, Fiona, I think one of the main reasons why our costs would have gone up this year was we had a dedicated event organizer, right?
Yeah, so
Yes.
they actually, I think, kept costs down for us in reality. So I think that they actually helped us budget much better. And they were, we had 3 different event managers that were interviewed, and they were the least expensive, and also recommended, in fact, by 2 patients who had actually used them before.
Yeah, no, I agree. I mean, I think it's fantastic having an event organizer. I think, yeah, my point was that in previous years, it was basically all of us running around trying to get things done in a less than organized way. So it's lovely to actually have an event organizer that was helping manage things. But yeah, I think overall, considering how much funding we actually had coming in, they were economically efficiently run, I thought. Any other questions for any of the board of directors?
Sorry, I got one more if that's all right.
Of course.
No worries. Are we attending the blood this year, the annual Australia New Zealand Haematology conference?
That's a very good question. Peter, do you want to make a comment on that?
I believe the meeting was in your city, Hasib.
The 2025 meeting was indeed. Yes, so I mean, this year the meeting was in our city, and I attended. But I think, Keith, is your question about whether the AAN is going to have a formal presence at next year's blood meeting?
Yes.
Yeah,
Yes. Sorry.
Yeah. I think that's a really fair question to the board, and I think that's probably something we as a board should discuss. I think it makes sense to have some kind of a presence at a meeting like that to really get the word out and expand our footprint. Peter, Fiona, any of the other board members like to comment?
I guess it depends what you mean by presence. I haven't been involved in organising the blood meeting for a number of years now, but to have a booth costs about $8,000. So we have to think, do we want to pay $8,000 to have a physical presence there? And we can get the actual price, but it's quite expensive. That's a discount for a not-for-profit compared to pharma, who pay, quite a lot more, typically more than 50,000 farmers paying. So we can get a quote on what it's like, but it won't be cheap. So a lot of these things you got to, weigh up.
I think doctors don't often visit booths either, and I think our impact is better felt through the research work and our education and support for the doctors and nurses who attend.
Yeah.
Bang for your buck that way.
We can get a quote and think about how worthwhile it is. We have managed to always, at the blood meeting, infiltrate the academic program, though, which has always been very important in terms of educating about amyloidosis to our doctors.
Yeah, and I can attest to the meeting this year. We had Matthew Rees, who we invited to present on amyloidosis, and he did have a little shout out for the AAN. So yes, I think that's a good way of getting the word out on the AAN.
Great, thank you.
No worries. Okay. Any other questions to the board of directors before we move on? I think if there are no other questions, then we'll move on to the next item on the agenda. And it is my great pleasure to introduce Muhammed Sabdia to present to us this afternoon. Muhammed is a PhD student from the University of Queensland, and he was recently awarded a Snowdon Foundation Gunn Family Fellowship Award for his project entitled Personalized Immunotherapy for the Treatment of Light Chain Amyloidosis. And so please welcome Muhammed. Thanks for joining us, and I'll let you take the floor.
Lovely. Thank you very much for that introduction. Can you see my screen?
I think we'll just get Libby to unshare and then hopefully yours will come up. Perfect. Yes.
Fantastic. Lovely. All right. Thank you for this opportunity to talk about some of my work, which, as Hasib mentioned, is about developing a personalized mRNA vaccine for patients with AL amyloidosis. And this work is funded through the Gunn Family Hematology Fellowship by the Snowdon Foundation. So I thought I'd just take this opportunity to walk you just through a little bit of the background and rationale behind this project. So there's not going to be any heavy data today, just hopefully a little bit on the story and the science. And I hope to answer a couple of questions just to frame this presentation. It's just going to cover a little bit about who we are, or who I am, and what is AL amyloidosis and why does it matter, and what exactly is a vaccine, because I know there is, before I started my degree, I had no idea what a vaccine was. I just know you get them at in the winter time to not get the flu. And then how can we really vaccinate against AL amyloidosis, and how do we do that? And then I guess summing it up in the big picture and where this could hopefully lead. So firstly, a little bit about who I am. So I'm a born and bred Queenslander who is almost a PhD graduate. I've submitted my thesis and it's under examination. So I'm very hopeful that I should be graduating early next year, which would be fantastic to get that off my plate. So I'm currently working as a Snowdon Foundation Fellow at the Mater Research Institute at the University of Queensland. And I'm working within the Blood Cancer Research Group at Mater Research. And we're based in this lovely building in Brisbane called the Translational Research Institute. And this building houses groups from the leading biomedical research institutes in Brisbane, as well as startups, and really facilitates a collaborative environment for developing translational research. And for this project, I'm especially fortunate to be working with Peter Millay, who is right next door to us at the Princess Alexandra Hospital. I'm also very fortunate to be working in a research group that shares the same aims and the same goals that I do. And I guess our overarching aim and goal is to become global leaders in personalized immunotherapies for Australian patients with blood disorders. And we know that Australians usually lag in access to new novel therapies, and our goal is to identify and develop these therapies locally. And we do that in the identification by identifying what makes a patient's disease unique, and what about their unique disease can we target. And on the development side, how best can a patient's unique disease be targeted? So that's who we are and what our goals are. And I guess I'll take this opportunity now to just briefly go over light chain, or AL amyloidosis, which really is the focus of my work. So AL amyloidosis occurs because of an abnormal plasma cell. But what is the plasma cell, and what goes wrong that causes this disease? So plasma cells are a part of our immune system that live within our bone marrow. And they produce proteins called immunoglobulins, or antibodies. And these immunoglobulins or antibodies are made up of two identical sets of the heavy chain, these really long ones here, and the light chain, which is this small fragment on the side here. Now, under normal circumstances, they help the immune system fight against infections and cancers and anything that's really foreign to the body. And what's really interesting about these immunoglobulins is that the plasma cells make is that they have a barcode. And like the barcode on a bag of chips, this barcode is unique to a plasma cell. No other cell in the body has this barcode, just like no other brand or flavor of chips has the same barcode. But what this means is that also patients don't share the same barcode. So every person has different barcodes within their normal plasma cells, and we have a whole bunch of these, tens of millions of them circulating within our body. But what this means is that we have a way of identifying which plasma cell makes an immunoglobulin or an antibody. Now, in the context of AL amyloidosis, there's a there's an abnormal plasma cell that lives within the bone marrow amongst the normal ones. And this plasma cell also makes immunoglobulins, but there's something defective in the light chains that it makes. And instead of making nice pairs with the heavy chain that the normal ones do, these light chains are sort of defective, and they make their way into organs such as the kidneys and the heart, where they accumulate and form clots, and we call these amyloid fibrils. Now, these fibrils really stress out the organs and cause them not to work and eventually shut down. And it's this organ damage caused by these amyloid fibrils that makes AL amyloidosis so dangerous. Now, when it comes to treating AL amyloidosis, there is one goal: stopping and or clearing the amyloid fibrils. This means treatments target the abnormal. Treatments can target the abnormal plasma cell that causes these amyloid fibrils, which should stop new amyloid fibrils from being created. Or you can directly target and clear the amyloid fibrils themselves that are already deposited within these organs. Now, currently there is a lot of work on removing the amyloid fibrils. However, there are no approved treatments, and the vast majority of current treatments focus on getting rid of the abnormal plasma cell, which is also the focus of my work. Now, the really interesting thing about these treatments is that they primarily come from treatments for myeloma. And just briefly, myeloma is a cancer where plasma cells grow uncontrollably within the bone marrow, and which means that treatments for myeloma are very, very good and effective at getting rid of plasma cells. So that's why they're applicable in AL amyloidosis. And this is just an overview of the progressive nature of treatments for both myeloma and amyloidosis. But the one thing I want to highlight is there's a clear lag between what's available for myeloma and when it becomes available for AL amyloidosis. And patients in Australia also get them later than our overseas counterparts. And this is one of the reasons I have such an interest in AL amyloidosis. I want to bring cutting-edge personalized treatments to Australians with AL amyloidosis. And the way that I'm interested in doing that, along with the Blood Cancer Research Group, is through immunotherapies, which have really dominated the past 20 so years of treatment. Now, what is an immunotherapy? Simply put, an immunotherapy is a treatment that assists the immune system. And this includes drugs, CAR T cells, and vaccines, which are all designed to naturally boost our body's defenses. But what is the role of the immune system, and how does it do this? So our immune system is a tightly controlled system that's really constantly working to protect us from the bad effects of abnormal or cancerous cells, or cells that have been affected with a virus like the common cold or COVID. And our immune system is able to do this because of the changes in these cells, because these changes are new. They're different to every other cell in the body. And this is what our immune system can recognize. The same way that we can tell if there's a weed growing in a veggie patch. It's new, we don't trust it, we've not seen it before, let's get rid of it. That's how our immune system works. Now, obviously, our immune system doesn't have eyes, so it recognizes these abnormal or cancerous cells through proteins that are unique to these affected cells, and these are known as antigens. So in a healthy cell, they have healthy antigens, and we call these self-antigens because they are of ourself. And in the context of a virus-infected cell, they have viral antigens, which are antigens from the virus. And in AL amyloidosis and cancers, we have what are known as neoantigens or new antigens, which are completely foreign to the body, that we've never seen before, and that don't exist anywhere else within our bodies. Now, in the context of AL amyloidosis and other blood disorders, these neoantigens can come from changes to the DNA sequences of the abnormal cell. And these DNA mutations make new proteins or new weeds that the immune system can pick up and identify. Now, on the left here, we see a normal blue DNA sequence with its normal blue protein. That's its self-protein. And now in the context of AL amyloidosis, these red changes in the DNA result in a red protein. And this red protein, which is called a neoantigen, it acts like a beacon for the immune system. So here it is on the AL amyloidosis plasma cell. It's just up here at the front. And this acts as the beacon for our immune system to recognize. And it's that weed that's hiding in the veggie patch. And it's important to note that these neoantigens, or these new antigens, also include that individual light chain barcode that's unique to each patient and to the affected abnormal plasma cell. So neoantigens are not normal, and they are unique to each patient, but they have the ability to stimulate an immune response. And they do this through special cells, which I'm going to call the educators. And the role of the educators is to identify the neoantigens and educate the rest of the immune system, namely the T cells. And these T cells are the real predators of the body. Once they catch a whiff, they hone in and they eliminate their target, which is this neoantigen expressing cell. But sometimes some cells really do slip through this process. And this is what gives rise to diseases like cancer or AL amyloidosis. And the role where vaccines come in is they're there to kickstart this process, essentially telling the immune system what to look for. Now, vaccines can target each of these 3 steps. And the trouble is, however, these methods of vaccinations are very good for common things like the flu or COVID, where a lot of people get them, so they can sort of justify the high manufacture costs, the highly involved manufacture, the rigorous quality control, and a very extensive market. But in terms of cancers and AL amyloidosis, these neoantigens are very, very personalized. So they're not really shared between patients. So it's not really cost effective, or there's no real market for the development of these methodologies for individual patients. So what we do need is we need a better method to enable a highly personalized treatment that specifically allows each person's individual immune system to target their individually abnormal plasma cell. And that's where mRNA vaccines come in. And I'm sure that we're all familiar with mRNA vaccines given their success with COVID-19. But I guess to really understand mRNA vaccines, they simply are just a vaccine made with mRNA, which I've just depicted by this little guy over here. But this mRNA is really fragile, and to protect it, it's coated in just a lipid sphere, which is a fat sphere, which allows it to be injected and taken up by the cells within the body. And now the mRNA used in an mRNA vaccine can be made to have the building blocks for many neoantigens in the one molecule. So this allows multiple neoantigens to be targeted in a single dose, so that when they're given to a patient, the cells make up the neoantigen, and the antigen-presenting cells, or the educators, eat it up, and they educate the immune system, including the T cells, so that the T cells can start targeting the cells that present any of the multiple neoantigens. And that's the benefit of mRNA vaccines, is that they're relatively cheaper than other personalized methods, and they can be produced much more quickly. And there's a lot less rigorous quality control in terms of testing, given that vaccines, they are effective, and we know a lot about that. And we're also very fortunate to be working with the base mRNA facility that's here at the same building in the Translational Research Institute on the PA campus. And this is a particular facility that has been built out of the University of Queensland, that has the expertise to synthesize GMP-grade mRNA vaccines, which is really promising for future translational work that can happen here at the VA Hospital campus. Now, this gets me to the last slide before I wrap up, which is just an overview of my project. And this work is all done in the lab. And we hope that the results of the study will be positive and allow us to move in a step closer to bringing these personalized therapies to Australians with AL amyloidosis and hopefully other blood disorders as well. Now, it starts off with the first and the most important step is getting the abnormal plasma cells from the bone marrow. And we're currently recruiting patients at the PA hospital, which is led by Peter Malay. And we're just obtaining material for our lab work. So unfortunately, there's no option to treat patients just yet, but we're hopeful that they can come in the near future. Now, the next step is to identify what makes these abnormal plasma cells different to every other cell in the body. We have to identify the mutations within the light chain and mutations elsewhere within the genetic code that we can exploit to identify these neoantigens. And these step 2 and step 3 have been the real focus of my PhD, and it's serving as a launching pad for this project. So I've spent the past 4 and a bit years working in chronic lymphocytic leukemia, which is a blood cancer, which is very similar in some aspects to AL amyloidosis. It has a very low mutational burden, so there's not many mutations in this leukemia and AL amyloidosis. And what that's allowed me to do is develop methodologies within the lab to identify with high confidence these neoantigens that are present within these abnormal cells. So it's building on the back of my PhD work that I'm really hoping to apply this to patients with AL amyloidosis, and I'm starting to do this within the next couple of weeks, which is very exciting, as we've already recruited 2 patients in the study. So this is just all built on my PhD work, and what really this grant is hopeful to fund me on is to synthesize and design the neoantigen mRNA vaccine. So this is a whole new world of science that I'm not going to really delve into because it's really complex and I don't really understand too much of it at the moment. But what this project is also going to allow me to do is figure out what the best way to deliver these neoantigens to patients and to activate our immune system responses. And once I'm able to synthesize this vaccine, this grant will also allow me the opportunity to test how effective they are in educating T cells and how effective these T cells are at actually killing the abnormal plasma cells that cause the disease. Now, I guess to bring it home, and I'm happy to take questions in a bit. So I've always felt like we as Australians, we need something that's our own. A lot of our drugs and innovations come from overseas. And I think we're really fortunate here at the PA Hospital campus to have access to incredible researchers like Peter Malay and incredible technologies that hopefully will allow us to set the stage for new novel therapies for not only patients with AL amyloidosis, but patients with blood disorders and even other malignancies. So I think there's a real space for novel therapies for Australians with AL amyloidosis, and it's my belief that mRNA vaccines can be an effective platform to give individual patients individual vaccines for the individual disease. So it's really bringing things personalized. And it's my hope that this will hopefully one day improve the outcomes of patients with AL amyloidosis. They can have their routine biopsy, get the vaccine design, and receive the vaccine all within a couple of months. And it's my goal to make this available to all Australians with blood disorders and other malignancies. And that's something that we're striving on in the Blood Cancer Research Group. And lastly, I just want to take this opportunity to acknowledge everyone in the Blood Cancer Research Group, the incredible clinicians in the amyloidosis clinic at the PA who are helping me recruit patients, as well as, and most importantly, the funding from the Snowdon Foundation and the Gunn Family Foundation. And I'm happy to take any questions.
Thank you so much, Muhammed. What a fantastic presentation and such exciting work in this field. I will open the floor to questions, and I've got Keith who's got his hand up that I can see, so I'll let you take that away. And if anyone else wants to ask questions on the chat, I'm happy to have that open. So I'll keep my eye on the chat if anyone else wants to ask questions. But over to you, Keith.
Thanks, Hasib. Let me just start off with Muhammed. Like, fantastic work you're doing. I had amyloidosis myself. I went through a stem cell transplant, so this sort of news is very exciting for me for the future. I suppose, like, I'm not a doctor of any sort at all. I'm a mechanical engineer. So my question to you is, with the Moderna vaccine that you're looking at, would this be something like a 3-month or a yearly vaccine that patients would have to get, or is this the idea is to sort of a one-stop jab and away we go, we're good to go?
That's a very good question. And the truth is, I'm not a clinical doctor, more on the research side of things, but my understanding of how that will work is we just have to try. So with the way that the mRNA vaccines are developed, we can make them one time, we can freeze them down, and then we can assess how patients are doing. They can receive the first dose. We'll have to obviously assess if there's multiple doses that are needed, like a booster, like COVID-19, for example. But the truth is, at this point, we don't know yet. I guess the whole point of the study is to show that it can actually stimulate those T-cell responses. And once we can show that, we'll have to assess how that works in people, in patients. But it should hopefully be something that works pretty well. But I guess time will tell, unfortunately.
No, thank you very much. Again, thank you for all the work you're doing with it. So good luck.
Thanks.
Kathy, I can see your hand up. Go for it.
Hi, Muhammed. It's fantastic work you're doing. I, like Keith, have AL. I'm the national, the patient advocate on the board. I was just interested, how can we as the AAN help you recruit patients to try and get more patients? Is this something only limited to Brisbane, or can we get bone marrow samples sent to you, or is there anything? I have no idea. I haven't spoken to Peter about this or anything, but I was just wondering, how can we help you recruit patients, because you only have 2 at this stage? Is that correct?
Yeah. So we've only been open for a few months here at the PA. It would be great to maybe talk offline about that, but there's a lot of regulatory and contractual issues that take months and months to get samples from anywhere. It took me about 6 months to just open up here at the PA hospital, and I'm trying to open up at a private clinic that's around here as well. But there could absolutely be a possibility to do that, and I think maybe Peter, being an expert in this, may know. But that's something that we can potentially have a look at.
Thank you. And thank you for all the work. It's just amazing. Like Keith, it gives us that are in remission hope that if we, when or if it comes back, that we have a lot more hope. So thank you, Muhammed. It was just amazing. And I hope that we can somehow put your speech up on the AAN website, if that is possible. Once again, I'll have to go through media and communications, but thank you.
Thank you.
Muhammed, can I just ask a question? Are you only looking at plasma cells from patients with AL amyloidosis, or are you looking at myeloma as well? I mean, obviously there's just a hell of a lot more myeloma patients, and the clones can be a little bit different but also similar. So
yeah, that's a very good point. So I'm working preliminary data up on myeloma and a few other plasma cell disorders, I think, like MGUS, for example. So that's data is being generated at the moment. So that would work as sort of a platform to I guess optimize these workflows, and then we can deal with apply them directly to AL amyloidosis, which obviously doesn't have many plasma cells and they're a lot more fragile. So I am doing work on myeloma and MGUS at the moment, and that should hopefully finish up early next year. we can get something out on that. And there's been some very interesting mRNA vaccines, or just more so vaccines in myeloma that are coming up nowadays, along with the CAR field. So I think it's a very hot button issue, and I'm obviously absolutely looking at myeloma as well.
Yeah. Okay. Any other questions for Muhammed? I think if there aren't any other questions, just on behalf of everyone on the line and the AAN as well, thank you so much, Muhammed. That was a fantastic presentation. We all really wish you every success in your research, because you know, it's going to mean a lot for the patients across Australia. So thanks so much.
Absolutely. Thank you very much to all of you, and thank you for having me speak here. I look forward to updating you one day with some good news.
Okay, thank you, Muhammed. All right, well, that was the final agenda item for the AGM. That brings our AGM to a close at 10 minutes past 5 Western time and 10 minutes past 8 Eastern time. I'm sure you're all keen to get on with the rest of your evenings. So unless there are any other comments, I just want to thank Libby again for all the effort and time that she put into organizing this and making it happen. She really does a lot to make these meetings run as smoothly as possible. So thank you, Libby. Thank you to everyone for attending, and I'll let you get on with the rest of your evening.
Thanks, Asib.
Thanks, guys. Bye.